or not, we hope you find this information helpful. We ask that you keep your microphone on mute for the duration of the presentation and put any questions related to this topic in the chat so they can be addressed accordingly. If you do have any questions outside the material being presented, please email support@ppsystems.com or asktheexperts@ppsystems.com, and we'll be happy to address your questions after the webinar. Both of those email addresses are available at the start of the webinar in the chat as of right now, and they'll be made available in the follow-up email I send out with the link to the webinar recording. So please allow me to introduce Dr. James Bunce. Jim actually built his first photosynthesis system as a graduate student 48 years ago and has been performing leaf gas exchange measurements ever since. He recently retired from a 40-year career with the USDA ARS in Beltsville, Maryland, as an environmental plant physiologist. His most recent work involves predicting impacts of global change on plant physiology. He's worked with many brands and models of photosynthesis systems over the years and prefers the CIRAS-3 Portable Photosynthesis System. So much so, in fact, that in his retirement, he's been working closely with PP Systems with our development and engineering team, testing and developing instruments, as well as providing technical and application support for all of our customers. So Jim, I'll let you take the floor.
Okay, thank you, Carrie. I'm Jim Bunce. We're going to talk about, the topic today is step-by-step automated tracking of diurnal patterns of leaf gas exchange in the field with the CIRAS-3 Portable Photosynthesis System. And I'm James Bunce, and as Carrie said, I retired from ARS and I'm working part-time with PP Systems, helping customers sometimes and helping design new gas exchange systems. So the first question is, why would we want to do this? Why track diurnal leaf gas exchange? And I have three reasons for that, two of which are shown here. First of all, we might want to try and understand what environmental factors are limiting photosynthesis at different times of the day. For example, Ph.D, leaf temperature, vapor pressure deficit, that kind of thing. Second reason for tracking diurnal gas exchange would be to determine the best time of day to compare treatments or genotypes or whatever comparison we're trying to do. Are we looking for maximum rates of photosynthesis? Or are we looking for stress tolerance at times of day when they're under stress? In order to figure out the best time to do this, we need to know what the diurnal pattern of leaf gas exchange is. A third reason for maybe wanting to track diurnal gas exchange is the example of holding the leaf conditions constant and see if the changing environment around the shoot affects the rates of leaf gas exchange. And here we're probably talking about problems with diurnal shoot water deficits, either in response to vapor pressure deficits or decreases in leaf water potential through the day. So those are some of the, and I want to give some examples of why those considerations are important. First of all, talking about limitations of photosynthesis through the day. This is just a diurnal curve of leaf photosynthesis. Starts out in the morning fairly cloudy, then the sun comes up, simulation rate bounces up, but then it has a slow, continuous decrease for many hours during the day. So the question is, what's causing this decrease over time? And we look at stomatal conductance, and yes, we see that stomatal conductance starts out low, comes up with the sun when the sun comes out, but then it also has a fairly strong decrease through the day. So that's probably partly involved in what's going on with photosynthesis. So what causes the stomatal closure? Probably it's at least partly because of an increase in vapor pressure deficit, which starts out at around 2 kilopascals and is going up to a little over 4. So that's a big change in VPD, and those are high numbers for VPD also. So that's probably what's causing at least some of the stomatal closure. And the changes in VPD, during the day are, as usual, caused primarily by changes in leaf temperature, which is going up from sort of like 25 when the leaf comes into full sunshine, and then it's going up to over 35, 36. So that's a big change in leaf temperature during the day. So we're probably, it's going to be CO2 supply that's limiting photosynthesis. And here we see when the sun popped out here, our CI settled down at around 200. But then as photosynthesis decreased, because CI decreased fairly strongly from 200 down to a little over 100. So that's a big change in CI, and that could fully explain what's going on with photosynthesis. And we just need to plug these CI numbers into a photosynthesis model to see how well it predicts the change in photosynthesis. And part of the change probably could also be related to the big increase in leaf temperature. So, so that's one example of using diurnal curves. Another one is looking at the question of what's the best time of day for treatment or genotype comparisons. And here's just an example for this. Again, sort of similar with light sort of popping, clouds going away and the light popping up. And this is photosynthetic rate of a leaf measured at ambient CO2. And you can see it start again like the other one. It starts, pops right up, and then it slowly decreases over time. Whereas if you compare that with a leaf from the same kind of plant, but measured at elevated CO2, so assimilation rate measured at elevated CO2, we see the same pop up, but here it's flat. It's not decreasing over time as it did in the ambient CO2 condition. So if we made the comparison at 11 o'clock in the morning, the elevated treatment has a rate that's 1.38 times the ambient rate. But if you made the comparison at 1 p.m., the elevated would be 1.76 times the ambient rate. So which number do you want? So if you wouldn't know unless you had done some diurnal curves that you could get such different answers at different time of day. And then you have to make a decision about when you want to do this. So the best time of day, knowledge of daily patterns is fundamental to designing comparisons among treatments or genotypes. Is your interest in the effects of maximum rates of photosynthesis, daily sums of photosynthesis, or the maximum differences between your treatments, whatever they are, genotypes or treatments? The third consideration is, this is just an old example of mine for a leaf held at constant conditions and the shoot being exposed to diurnal variation, and we get a midday stress. And just to illustrate here, photosynthesis here is down on the bottom curve. The open symbols, this is a corn leaf out in the field. And you can see 8 o'clock in the morning, it has a rate of around 55, which is perfectly happy. But then it's decreasing all the way till 2 o'clock in the afternoon. And that's a fairly substantial decrease. Stomatal conductance is not decreasing. Stomates are not closing. This is, just a small little bit of an increase in stomatal conductance, actually. And here we can, up the top figure, you can see light is doing the normal sine wave. And leaf temperature is fairly moderate, going from like 24 up to about 30. So it's nothing extreme. So in this case, what's happening, turned out that it's a diurnal drop in leaf water potential is causing this reduction in photosynthesis. So again, another reason for tracking diurnal patterns. In this case, the leaf was held at constant conditions and the external environment is forcing the leaf to do different things. So my message about this, of why do diurnal patterns, is that the knowledge of diurnal patterns is fundamental to designing comparisons among treatments or genotypes, especially in the field, but not only in the field. These kinds of patterns happen in greenhouses. They can happen in indoor controlled environment chambers, even under constant conditions. You can get a diurnal change in gas exchange. And do the comparison you want to do for whatever study you're doing, it's advisable to know what those patterns are so you can make an intelligent choice about what time of day to do these comparisons. Okay, so hopefully I've convinced you that it's worth doing these kinds of things. So how do you do it? How do you measure diurnal patterns of leaf gas exchange in the field with a CIRAS-3 instrument? You can do it the way I did for many years, and that's do it yourself manually. You can have, if you have students or helpers, it's easier to have them do it than for you to stand out there all day and do it. Or you can automate the process. And automating the process is what I'm going to go through here. A warning, first of all, about the CIRAS-3 and water. And this applies to all portable photosynthesis systems. Neither the console nor the leaf chamber are waterproof. Neither is the base of the console. You don't want it to sit in a mud puddle. If rain is possible, be prepared to quickly stop your measurements to save your instrument. Getting these things wet is not a good idea. It's going to cause you a lot of downtime. And I have lots of funny stories about scrambles to get out in the field for unexpected rainstorms. I've done it many times. Another message here is never run the inlet humidity at 100% of ambient if you're not going to be there next to the instrument, because at times the environment can change suddenly. And when your gas exchange is going fine and the environment changes quickly, you can very easily get into a problem of condensation inside the leaf cuvette. If you run into the condensation inside the leaf cuvette, that takes a long time to dry out and correct itself. So you're essentially done for the day if that happens. It takes a long time to get everything dried out again and up and running again. So the warning there is just never run it at 100% of ambient unless you're going to be right there. So those are some warnings. All right, so question of how to power the thing. And to be honest, I have always just used line power. I don't have experience trying to operate for a long period of time just on the battery, even with no lights. And the process of tracking ambient temperature can require quite a bit of power in full sunlight. And if you were trying to run with constant light, that would increase your power load even more. Two standard batteries in the CIRAS-3 give up to 12 hours, but that's probably not when you're trying to control temperature on a real bright sunny day. There is a supplemental battery pack that you can add to the two standard batteries, but I don't have experience with trying to do it that way. As I said, I always just use line power. My field plot has line power there. Some other considerations, shading the console. This is just a matter of orientation. You want to shade the rear chemical columns to avoid them overheating. You want to shade the screen to avoid it from overheating. These are things that I typically do when using it manually in the field. I'd run the instrument with the sun at my back in order to shade the screen and the chemical columns and stick the leaf cuvette out in front of my shadow as I'm doing measurements. So these are things you need to do even if you're not there. So shading the console, just a white styrofoam board clamped to the handle, then you can use the handle pivot to adjust the angle of that very easily. And that works fine. The chamber, leaf chamber, has an air temperature sensor, which is going to use to control your temperature usually. And that temperature sensor is right here under the base of the handle. So that can heat up in the sunlight. This whole metal, aluminum, whatever the metal is, shiny, silvery handle heats up in the sunshine. So you need to shade as much of that handle as you can, but you don't want to shade your light sensors, the external or your internal light sensors. So one way to do that Cuvettes you can use with a tripod, but for field and shading, I tend to use traditional ring stands because that gives me a place to anchor my shade board. And I just put a weight at the back end of it and just dangle it over the back end of the leaf chambers, the handles of leaf chambers. You can see two of them set up here on a soybean plant very early in the morning. So another consideration is CO2 supply and airflow rate. If you're using a CO2 cartridge, which is normally the way, normally most appropriate and the way it's recommended to do things, if you can do that, the CO2 cartridge has a limit. And these are just some things that I have learned. So there's nothing scientific about this except experience. And that is if you're running it 400 parts per million with a cuvette flow rate of 300 cc's per minute, that'll work fine for 24 hours. So you can do a complete 24-hour curve if you want to at 400 parts per million and 300 cc. At 400 parts per million, 350 cc per minute is sort of marginal for 24 hours. You may run out. 12 hours, it would work fine. If you higher CO2, you're going to use CO2 faster. For 600 parts per million and 350 cc's per minute, that will work fine for 12 hours, for a 12-hour day. For 600 parts per million, 250 cc per minute, CO2 supply will last for 24 hours. So these are just some things to get you in the ballpark of what you can do. If you're using ambient air in the field, you probably should be using a sampling pole, which PP Systems provides as an option. And especially if you're trying to get gas exchange rates at night, you're going to, even with a sampling pole, you're going to need a big buffer volume to dampen out CO2 fluctuations, because at least in my field situations, the CO2 concentration at night just depends on puffs of wind coming through, and it can change very drastically in a short amount of time. So you're going to need a bigger buffer volume than you're used to needing to use if you're trying to do this at night. Okay, humidity settings for tracking ambient air temperature. This is again a matter of getting some experience with your plant material. You have to have some idea of what your leaf's maximum stomatal conductance is going to be in the middle of the day. And you're going to need to adjust your percent of reference air as you would if you were doing midday gas exchange measurements based on your flow rate, your window size, and your maximum stomatal conductance. Normally, when you're trying to measure under ambient conditions, your target is to have the sample water coming back from the cuvette equal to whatever the outside ambient water content is. So that's your target, but it's a matter of knowing what your flow rate is, your window size, and your maximum stomatal conductance as to how you need to set your percent of reference incoming humidity. Logging of data. Basically, you have unlimited storage. So whatever frequency you want, you can have. I often have used storing data every 5 minutes, which gives you 288 data records in 24 hours. If you're using more than one instrument, say setting them at 2 different CO2 concentrations, which I do sometimes, it's worthwhile to take the step of synchronizing your clocks on the 2 instruments so that it's easier to put the data side by side. Programming tips from the settings menu of your CIRAS-3, and I'll show you a screenshot of that in a minute. You're going to be using automatic zero and differential balance as one of your settings so that the instrument can do its checks whenever it feels the need. You're going to set the approximate reference CO2 unless you happen to be using ambient air. But normally if you're using a CO2 cartridge and controlling CO2, you're going to use the approximate reference CO2. And if that doesn't get you as close to your target as you want, there is a way to, there's a function in the CIRAS-3, which is called find max C. And if you do that function, then your reference CO2 will be closer to actual CO2. So your reference will be a closer approximation if you use that find max C function. You're going to set the percent of reference humidity, and we talked about how to figure out how to do that. And your setting is going to have no lamp unless you want to have a lamp, but normally you're using sunlight. You're tracking sunlight. And usually you're going to track leaf to the ambient air temperature. There are times when you might want it to be constant, but tracking diurnal patterns, you just track leaf to ambient air temperature. And then you just do a timed recording and you pick your time interval. So what the CIRAS-3 window looks like, this is the settings menu. You're doing gas exchange, you have whatever cuvette you have. This is the 18 millimeter diameter window. And as we went through, your zero differential balance mode is going to be automatic. Your reference CO2 reference is going to be approximate reference air, and you pick the value. Your water reference is going to be a fixed percent of reference, and you pick your value. Your flow rate, cuvette flow rate is whatever is appropriate for your leaf. Analyzer flow, I just leave that at 100, which is normally the way it runs. You're going to, in this case, using ambient light source. So it's just going to use sunlight. So you don't have to worry about color of light. Your temperature sensor measurement, I fill my cuvette windows with leaves. So I use the infrared sensor. And in this case, I'm controlling leaf temperatures so that the leaf temperature is tracking ambient temperature. And your leaf area, your boundary layer conductance, resistance, and your to metal ratio. So that's what your settings window looks like. So it's really easy to set all these things up. And the last thing I want to give you a little bit of a warning about, as in Maryland, as in lots of other locations, leaves often have dew on them for a few hours after sunrise. And you don't want to enclose a leaf that's wet with dew. You're going to get nonsense if you do that. So you can dry them with tissue and let them air dry a little bit longer than that before enclosing. Or as an alternative, you can wait a little longer in the morning until they dry and then clamp them in and get your early morning values the next day if you need early morning values for what you're doing. Okay, and that's it. I'm going to open things up for questions.
Well, thank you very much, Jim. That was terrific. We've got some questions here. First question, is it necessary or do you recommend to stop the automatic differential balance while measuring diurnal variations?
It'll displace the time a little bit, but it's not, your stored data is going to be fine. It's not going to affect your stored data at all. It'll affect the timing of the storing of data a little bit, but not enough to worry, not enough to bother you.
Okay. It looks like that's the only question right now. Does anyone else have any questions they'd like to ask Dr. Bunce?
Hi, this is Francisco Arriaga. I'm with the University of Wisconsin-Madison. Thank you for your webinar, your presentation, and sharing all your knowledge from all your experience. So that's really good to see. I have a question related, I guess, to instrument. I realize that the webinar is sponsored by PP Systems, but any thoughts on, there's other devices out there and anything's pros and cons and One thing I'm also interested is looking at maybe CO2 emissions out of soil, what capabilities, are particular, I guess, to the PP system, but any thoughts on that in your experience?
The PP systems does have an automated soil CO2 flux
I'm
system.
sorry, can somebody mute that, please? Jim, you're muted now. You need to unmute yourself.
All right. Yeah, PP Systems has automated soil CO2 flux system, which has some advantage over the competitors. And primarily in that the systems are not tethered to one central location, so you can have them independent and remote from each other without any trouble. And it's set up to record 24 hours a day at whatever interval you want. So for soil CO2 flux, that works fine. In terms of, I don't have experience trying to do diurnal curves in the field with other photosynthesis systems. I know. So I'm probably not the one to ask that question of. I've done it with PP systems, and I've done it with homemade systems, but I've not used other competitor systems that way.
Yeah, I've used a competitor in the past, and it was cumbersome, but it was an older model too. I haven't done any recent, so I don't know if Carrie or Tim can kind of share some key benefits, I guess, of the PP system. I've used actually PP on a different setting.
Mm-hmm.
For CO2, and a colleague of mine used them heavily. So I know they had this open phase chambers where they were controlling the CO2
Yes.
Environment CO2 basically. And so they use a PP system. So I know they really liked it. So I don't know if you could share specifically, at least for the leaf type setup, what advantages the system has.
Well, certainly, there's a lot of advantages to the CIRAS-3 in particular. Certainly portability is first and foremost. The system is by far one of the most portable on the system, portable system on the market when compared to other systems that are out there. You can compare the technical aspects and technical specs, of the various systems. We offer a much greater temperature control range for doing temperature responses is another advantage. The infrared temperature sensor for leaf temperature, which is a non-contact measurement that measures over the entire leaf area. That's also a significant advantage compared to other systems that are using a thermocouple or leaf thermistor, which has a number of disadvantages as well. Most systems, at least, like PP systems or, LICOR in particular, a lot of the capabilities of the system, accessories for doing simultaneous chlorophyll fluorescence, soil CO2 flux, various leaf chambers for doing various leaf types or plant types. Those are somewhat similar, and finally price typically, where funding oftentimes is limited. Certainly we offer a very advantageous price when comparing to these other systems that are available.
Thank you. That sounds great. Thank you so much.
You're welcome. Jim, we've got another question here.
Okay.
If I have another type of cuvette instead of the, what I'm assuming is the PLC universal cuvette, and I want to measure photosynthesis in Arabidopsis, can it be cut faster to measure the leaves?
In other words, can you measure cut leaf photosynthesis on Arabidopsis? You can. Yeah, well, Arabidopsis is a funny creature. It actually likes to grow in high light. And if it grows in high light, like 500 or 600 micromoles, it develops leaves that are perfectly big enough to fill a cuvette window of a CIRAS-3 PLC. So there's no reason that you need to cut a leaf to do photosynthesis measurements in it. If you're dealing with Arabidopsis grown the way a lot of biochemists do at 100 micromoles or something, they're tiny. And there it's very difficult to get them into a leaf chamber. And there you might want to cut them. Measuring cut leaves in a photosynthesis chamber has been done for years. I've never been comfortable doing it. But the procedure is that you try and cut the petiole underwater to avoid air embolisms getting in. And then if you have the petiole inserted in degassed water and you've cut it underneath the surface of the water, supposedly you can get reasonably steady rates for quite a long period of time. It's not something, I've never tried it with Arabidopsis. I'm just assuming that based on what other people do with other species, you could get away with it. My preference with Arabidopsis is to grow them in an environment where you get leaves big enough to deal with routinely.
Okay, well, thank you again, Jim. I see we've got a few other questions that don't particularly pertain to the webinar, so we'll take those and answer those separately.
Okay.
We'll give one last chance. Does anyone else have any other questions for Dr. Bunce and the material that he presented today?
Well, as always, Jim, thank you for your presentation, and thank you to everyone for your great questions. If you have any additional questions related to this material, please feel free to reply to the email you received from me, and we'll answer you directly via email. For unrelated questions, please email support@ppsystems.com or asktheexperts@ppsystems.com. Both email addresses will be in the follow-up email you will receive from me with the link to this recording. Thank you so much for your time today, and I hope this was helpful. Thank you. Thank you for attending this webinar. To learn more about how the CIRAS-3 Portable Photosynthesis System can enhance your research experience, visit ppsystems.com.